What's up?
So to explain the mechanism of ruxolitinib, we have to recall what is JAK2 kinase and how mutation in JAK2 kinase affects hematopoiesis.
We got to take care of our immortal souls.
You know you can't read.
It's the Bible, you get credit for trying.
On chromosome 9 located gene which is called JAK2 gene. JAK2 gene encodes JAK2 kinase protein. Basically, JAK2 gene on chromosome 9 is a number of nucleotides that form DNA molecule. To use information in the gene, information must be carried to ribosomes, where proteins are made. So DNA molecule for this purpose makes a copy of the gene in form of matrix RNA molecule. And then matrix RNA molecule is delivered to ribosomes. Ribosomes read matrix RNA molecule in codons and include complementary to that codon amino acid. In this case, it's valine and glycine. And valine here is located at 617 position. And a particular amount of amino acids form protein. In our case, this protein is JAK2 kinase. The function of JAK2 kinase is to activate cellular pathways that are responsible for proliferation of myeloid cells. And activation of pathways occurs by phosphorylation. In order to activate pathway, JAK2 kinase use ATP molecule. By ATP molecule, it phosphorylates proteins in this pathway, and as a byproduct, ADP is released. By activation of JAK-STAT pathway, JAK2 kinase stimulate mostly red blood cells production. By activation of PI3K AKT pathway, it stimulates production of megakaryocytes, from which platelets are formed. By activation of RAS pathway, it stimulates production of granulocytes and monocytes. And in normal condition, JAK2 kinase is active only when it receives signal by cytokines. Kidneys produce erythropoietin that stimulates red blood cells production. Liver produces thrombopoietin that stimulates production of megakaryocytes, from which platelets are formed. And bone marrow microenvironment, by secretion of granulocyte macrophage colony stimulating factor, cause increase in production of granulocytes and monocytes. But if mutation cause substitution of valine on phenylalanine in 617 position of JAK2 kinase, now JAK2 kinase becomes constantly activated. So this regulation by cytokines is gone. And this mutation causes disproportional activation of intracellular pathways that results in mild increase in granulocytes and monocytes, moderate increase in platelets, and severe increase in red blood cells.
Now what?
To decrease the effect of this mutation, we use ruxolitinib. Ruxolitinib blocks the ATP binding site on JAK2 kinase. So now ATP molecule cannot bind to JAK2 kinase. Without binding of ATP molecule, phosphorylation becomes impossible. And without phosphorylation, proteins that are responsible for proliferation of myeloid cells cannot be activated. Thereby the production of granulocytes and monocytes will decrease, also the production of megakaryocytes and thereby platelets will decrease. And in addition to this, the production of red blood cells will decrease. So with time, ruxolitinib provides normalization of blood cell count. Simple as that.
I like it. Simple, easy to remember.
And because mutation in JAK2 kinase is involved in polycythemia, essential thrombocythemia, and primary myelofibrosis, in all these disorders, ruxolitinib can be used. And actually, nowadays, it's the most effective drug in treatment of all these three disorders.
Great. Fine. Yep. Ciao. What's that mean? Ciao. It's Italian. It means food.